Okay, so this is fluorescent microscope. Unlike other microscopes where we can get the bright field images, this can be used for multiple purposes because this is equipped with the bright field imaging system, phase contrast, as well as the fluorescent microscopy. Now, before you use it, you need to turn on this from the power button, which is here. Now once you turn on the power button, you can see there is a light coming up from the bottom source, and so this is on the objective, and you have to set your objective at 10x when you need to focus your specimen. So bring the objective at this point, and this is your eyepiece. This is your eyepiece. Set it, this eyepiece as well as the objective, and this is the Y knob that can be used to move the stays and front and back and both the sides. So, and after that you put your samples over here, a specimen in the that would be there on the slide, and move the knob to adjust it, and this is the fine course that can be used to further move the stays up. And then this is a fine tune which is adjusted over here in the main course. This can also be used for adjusting. Once it is adjusted, you can adjust the brightness of the image by changing the amount of light that is coming from the optical path. So if you move a little bit front, then the light source will get a little dimmer, while if you move it towards yourself, the light will be a little more brighter. And after that you can adjust and start watching from this, and while you are fine tuning your with this fine tune course. And once everything is adjusted, this is something that can be used to adjust the interpupillary axons, and so that if there are two cells, you cannot see them overlapping on top of each other. So this can be used for that purpose. Now once everything is set up and you want to capture the image, then this is a camera which is fit in, inbuilt with the system, and this camera can be turned on. First you turn on the camera from here, and this will allow you to capture the image from your system which is connected, and the system is connected with a software. Software is this NIS element D5.20.00, that's 64 bit, and this can be used to capture images right directly from your microscope. So that's for the bright field imaging. And if you want to take the phase contrast imaging, then you need to adjust at this one, at the pH1, and this will allow you to take the phase contrast image, pH1. These things are done. So these are two things that you can use. And if you want to take the fluorescent image, then there's another system which is sitting over here. You need to turn on the power. This is called mercury lamp, and this mercury lamp will provide the light source from the back side, and the light will come directly from that side to the top. And there are filters. We have 3 filters: DAPI, FITC and TRITC. DAPI, if you set it on 1, now the DAPI is on, and this DAPI will give you the blue light images. So if your sample is stained with the Hoechst 33342 dye, your sample is stained with the blue fluorescent protein, the images can be captured using this DAPI filter. And if your sample has the FITC dye, that is a green fluorescent dye, then that can be captured with this at the FITC channel. And if the sample has the red color in it, like Psi5 and red fluorescent protein, then you need to set at filter 4, and the TRITC will capture the images on the right side, on the right correct wavelength for the red images. Now these images can be captured, and I'll show you the one example when we are trying to collect what we have actually done. I collected the image in DAPI, and that we have done with the microscope while connecting with the system, and this is how the images will look like. So this, when you open the software NIS Element D and capture the images, this is how the blue light that comes in the DAPI filter, and one can capture the images. So there are various ways that the fluorescent microscopy can be used, and this will provide you a multiple added advantage, such as if you have to find out the subcellular localization of certain proteins, whether they are sitting in the nucleus or in the cytoplasm, that can also be visualized with this particular microscope. One can use the fixed sample with these things, and it can be used for the multiple purpose.